All right. Good morning, everyone. Thank you for joining us in the August version of Gene Delivery Seminar Series. I'm very excited to introduce our speaker for today, Connie. Dr. Connie Rich holds a PhD in Physiology, Development, and Neuroscience from the University of Cambridge, where she was a Cambridge Trust and Herschel Smith Scholar. She went on to complete postdoctoral research at King's College London and the University of Sheffield, focusing on evolution and development of the nervous system and limbs. Before transitioning into industry, Connie served as an assistant instructional professor at the University of Florida, where she taught anatomy and physiology and was recognized as the UF College of Liberal Arts and Sciences Teacher of the Year. Currently, Connie works as a scientific product manager and operations coordinator at Vector Builder, where she combines her expertise in molecular biology and education to create scientific content, coordinate product launches, and support researchers worldwide. And in today's seminar, Connie will do a deep dive on how to design and optimize AAVs for research and therapies. I'm very excited about this seminar, and Connie, ready when you are. I do want to mention that this session is being recorded, and if you have any questions, please put it in the Q&A box, and it will be answered at the end. All right, on to you, Connie.
Perfect. Thank you so much, Samita. And thank you everyone for coming. I'm really excited that we're expanding our seminar series to include not only kind of research innovations and perspectives, but also some educational resources that can really provide some guidance and some practical tips on how to use popular delivery systems, including AAV, as we're going to talk about today. So, because we have a very diverse, largely academic audience, including people who have never worked with AAV before, we're going to do a pretty thorough coverage of background, highlights, and comparisons for AAV, really highlighting why you should use this and when you should maybe use a different system. And once we talk about kind of why you would use AAV over other systems and really its practical use, we're going to go into some key design tips for creating AAV vectors that will hopefully do exactly what you want in your host cells. And we'll talk about some optimizations, particularly with regard to enhancing targeting and efficiency for your AAV vectors. And we'll wrap things up with some key considerations if you're thinking about taking your research to the clinic. So when you're delivering genetic material to your host cells, adeno-associated virus or AAV is one of the most popular options today for both research and clinical use. Way back in the 1960s, it was discovered as a virus-like particle that was found in adenovirus preparations, looking almost like a contaminant or an artifact. But intensive study has revealed that this is incredibly useful for research as well as therapeutic applications, partly due to its really beneficial safety attributes. So the AAV virus exhibits no pathogenicity, has very low immunogenicity. It doesn't trigger a strong immune reaction, and even the wild type virus, when it infects host cells, it's really poor at replicating inside of the host cell without help from a different virus. So when your AAV viral particle binds to receptors on the surface of the cell, for instance, heparan sulfate proteoglycan, it's endocytosed before it escapes from that endosome and is either degraded by a proteasome or is transported into the nucleus where it's uncoated, and the single-stranded AAV genome from the virus goes through double-stranded DNA synthesis before kind of transferring into this episome, this circle of DNA that remains outside of the host genome. So while AAV characteristically has no integration, I did include a little asterisk here because historically, generally, it is not an integrating virus. However, pretty recent studies have shown that you do have some small percentage of integration that can happen into the genome, which is something you have to consider when you're thinking about things like clinical applications. So With this episome right here, it's just kind of hanging out. If there is a helper virus, for instance, adenovirus or herpes simplex virus, then the proteins from that helper virus can help in the replication and packaging of new AAV variants. Without additional helper virus, this episome just remains latent in your host cell for pretty much the entire life of the cell. Now, the dependence on that helper virus is actually pretty beneficial for AAV because it means that the virus can have an extremely compact, simple genome. So AAV has only 4.7 kb in its viral genome. It's the single-stranded DNA genome of just really two protein coding genes or two protein coding regions that are flanked by these inverted terminal repeats or ITRs. They form this characteristic T shape thanks to palindromic sequences that form these B arms and C arms on either end, and then this A segment kind of in between. We're going to talk a lot more about those ITRs later because they play a really important role, especially when we're talking about using AAV for research or clinical applications. Now, in between those two ITRs, we have just really two genes that form a small number of proteins. You have the rep gene that's responsible for creating proteins that are important for replication and packaging. Again, they can't act on their own. In order to make a new virus, you need those helper virus proteins, but these are responsible for like the AAV specific replication and packaging. And then we also have this cap gene that creates proteins that are responsible for forming the capsid that encapsulates the viral genome. So this capsid contains proteins that bind to specific receptors on your host cell. And initially, the researchers found one particular AAV, AAV2, that's really widely found, and it binds to this HSPG, which is found on a wide variety of host cells. So it targets a wide range. However, we subsequently found a large number of other capsid structures that allow for there to be various different targeting mechanisms that allow for targeting of different cell types. So you have various different serotypes that are available for different types of AAV. So you have differential targeting as well. Now, excuse me. Now there we go. For AAV, as well as a variety of other viruses, we can take advantage of the really highly efficient infectiousness of the virus, but we can use it to overcome a significant hurdle in gene delivery experiments, which is actually delivering that DNA, RNA into our cells. So what we can do is we can remove the viral DNA and we can replace it with our sequence of interest, whether that's coding for a transgene or if it's just coding for an RNA. We can take everything between the ITRs out and just replace that with our own sequence. Additionally, we can select which capsid we want to use. So if we want to target if we want to target the brain, we can just use any of the cap genes from these various different AAVs to target our particular target cells. So once we create this recombinant AAV, it's going to enter the cell typical to wild type AAV, and it's going to be transported into our nucleus. There, it's going to be uncoded, and our single-stranded AAV is going to go through double-stranded DNA synthesis before creating the episome. Now, from here, it's going to use host cell machinery to create our transgene, or we can just create our RNA, and it can either be used in CRISPR or for knockdown experiments. However, if you want your RNA or your transgene much more quickly, there is a little workaround that you can use. Instead of encoding this on a typical wild type style viral genome, you can use not single-stranded DNA, but you can actually use double-stranded DNA. So you can have this thing self-complementary AAV. This self-complementary AAV is going to have the benefit of being transcribed and translated more quickly. However, because your viral genome still has to be 4.7 kb, it is going to have a pretty significant limitation in the amount of your chosen DNA that you can put in here. So you're going to end up with less than 2 kb of space that you can actually utilize. Comparatively, for a single-stranded AAV genome, you're going to have about 4.2 kb that you can really utilize. So to create this virus that is able to express our gene or our RNA of interest, but still have the safety of not potentially creating AAVs in our host cell, we still need those proteins that we've just removed. So we need not only the things that we want to be expressed, but we need proteins that are important for creation of this recombinant AAV. So to make this happen, what we need is 3 different plasmids. We're going to have the transfer plasmid that has essentially what we want to be expressed. So those are the things in purple that are going to be expressed in our host cell. Additionally, we have these green elements here that are kind of more viral specific elements. So this is going to be the inverted terminal repeats, as well as sometimes regulatory elements. Of note, we do, of course, because this is a plasmid, have to have these bacterial components in blue as well, our origin of application, and some antibiotic resistance gene. So in addition to this, we need 2 packaging plasmids. One of them is going to contain the rep-cap genes from the AAV viral genome. And this is going to be driven by a promoter and have a polyase so that these proteins can still function. But as I mentioned at the beginning, we do still need to have those helper virus proteins in order to create an AAV variant. So we deliver those on a separate plasmid that's just encoding for those adenovirus helper genes. So once we, forgot to mention this, we have our rep-cap genes from our AAV genome that we can select actually which rep cap genes we are using. So particularly those cap genes can encode for these different capsids depending on what we are targeting. So if we want to target, say, the inner ear, then we can choose AAV 2.7 or AAV 6.2. And if we're wanting to target the skeletal muscle, we can choose AAV 9 or AAV 1. And of course, we're going to just choose depending on our target tissue type and depending on the kind of level of acceptability for off-target effects, because there are very few AAVs that only target one particular tissue. We're going to talk about some workarounds for that later. So once we have our 3 plasmids, our transfer plasmid and our 2 packaging plasmids, we transfect these plasmids into our packaging cells. These are HEC293 T cells, and they create those AAV variants, those recombinant viruses. And then we can isolate and purify that AAV, and then we can just deliver it straight to cells either in vitro or in vivo. So while the kind of design and preparation process is pretty labor and effort intensive, once we have that virus, things get a whole lot easier. Now, by comparison, if we're using non-viral vectors like plasmids, the design is much, much easier. We still have those components that are expressed in our host cells, but we don't have the requirement of all of those other viral proteins. We typically don't require helper plasmids. And so the design aspect and the production process is significantly easier. However, introducing that non-viral vector to our cells is a lot more labor intensive. So we have to perform transfection with something like electroporation or lipofection, and both of those can be more inefficient, and they can also potentially cause damage to our cells. Now, there are times when these non-viral approaches are preferable. If you have easy to transfect cells like HEC cells, for instance, like we see right here, or CHO cells, or if you have a really, really large transgene, because plasmids can carry upwards of 30 kb very easily, or if production costs are a major concern, then non-viral vectors are a good option. Whereas viral vectors tend to be the better option if you have difficult to transfect cells like neurons or pluripotent stem cells or primary cells. Or alternatively, if you're using in vivo delivery, this tends to be the better option. There are, of course, exceptions. And then if you want tissue specific targeting, this allows for a much higher level of that. So if we decide viral vectors are the way to go, that's exactly what we need. We have many different options beyond just AAV. So how do we know which one we're going to go for? So when we're looking at popular viral vectors, these are our major players. And an easy way to divide them is whether you have stable integration into the host genome or not. So if you need stable integration, especially if you're working in vitro, so whether that's studying things through stable cell lines or, for clinical applications, doing ex vivo treatments for HSCs, then these two are going to be kind of what you're debating between, either lentivirus or our gamma retroviruses here. Now, for lentivirus, this tends to be what people go for because it does have a slightly higher carrying capacity. It has very broad tropism. But for both of these options, you do have the issue with potential for host gene disruption. If you're looking for something that doesn't stably integrate, especially if you're working in vivo, then you're going to be looking at one of these two options, our AAV or our good old friend adenovirus. So when you're debating between these two, AAV does tend to be by far the more popular option because although you have a much lower carrying capacity, the very low immunogenicity, especially compared to adenovirus, and the control that you have over the specificity makes it a really good candidate. Additionally, while AAV is transient, 11 asterisks here. While AAV is transient, because it remains as an episome, if you use this in non-dividing cells, it is going to allow for really long-term expression. So overall, AAV is a really great option if you're doing in vivo gene studies or if you're doing in vivo therapeutic development, if you're targeting very specific cell types, or if you want really long-term expression without genomic integration. So for instance, for gene therapies for neurodegenerative disease, AAV tends to be a really popular option. So once we have decided that AAV is our way to go, we're ready. Let's make a recombinant AAV and let's find the next biotech innovation. We have to consider the design. And of course, we're going to be looking at the design of these elements that get expressed in our host cell. But it's important to note that we need to keep in mind the design as well as the quality control of not only our transfer plasmid, but also our different packaging plasmids. So we're going to start, there we go, with our transfer vector. So for the transfer vector, the first thing that you're going to do for any new gene delivery experiment is you're going to make sure that these purple elements, these host cell elements, are designed to express the thing that you're looking to express in your host cell. Now, the first port of call for really anything having to do with your recombinant viral genome is size, making sure that from one ITR to the other is no more than 4.7 kb. And removing those ITRs as well as a couple of other kind of sequence elements here, that leaves you about 4.2 kb to work with. So when we're thinking about what to use, let's just start with the promoter because we have to drive expression of our thing. Now, if you're using something like AAV2 to cause broad targeting, you're going to want a ubiquitous promoter. And one thing that we have found really regularly is that when using CMV for in vivo delivery and you want as broad of expression as possible, CMV tends to cause some silencing. So you actually want to avoid that and instead go for something like CAG if strong expression is your primary goal, or EFS if kind of size is your biggest concern, because EFS is a very small promoter. Now, you can also have tissue-specific promoters, of course. So you can not only have serotypes that target specific tissues, but you can also use a tissue-specific promoter to enhance that specificity. However, it's a note that these are a bit more restrictive. Their efficiency, their level of expression varies depending on the promoter. And additionally, they do tend to be larger, which can be a problem with when you're working with a space so small. Now that ORF is going to be driving expression, sorry, the promoter is going to be driving expression of your ORF. And the first thing you want to do is make sure that the promoter is matching the ORF. So if you are expressing a transgene, you want to make sure that you're using a pull-to promoter, for instance, CAG or EFS, or for instance, that tissue-specific promoter. Whereas if you're driving expression of a short RNA, for instance, for a knockdown experiment or for CRISPR, then you're going to want to be sure that you are using a pull-3 promoter like U6. And one additional note for ORFs is that we have a pretty simple setup here, but even with AAVs, even with this restrictive space, you can have pretty complex vectors that allow for expression of multiple components. So you can have multiple ORFs that are separated by linkers, or you can even have an entire small cassette where you have a promoter, ORF poly A, and then another promoter, ORF, let's say an RNA, and then another poly A or another terminator. So this is particularly helpful if you're wanting to do a CRISPR experiment. If you're wanting to express both the Cas9 and the gRNA together on one vector, that is possible in this space. But it's important to note that you cannot do this with the kind of stereotypical Cas9, SP Cas9, because that is going to be too large to do this with AAV. So instead, you can use SA Cas9 from Staphylococcus aureus. And that allows you enough room to express not only SA Cas9, but also an accompanying gRNA. Another kind of quick note, that gRNA is going to be designed differently to be compatible with your SA Cas9. So once we have our promoter, our ORF, or even potentially multiple ORFs, we can't really stop there because there are other components on this transfer plasmid that need to be carefully designed and carefully monitored as well. So in addition to those host cell components, we do have these virus-specific components as well. And of course, we again need to be sure that everything is kept between those ITRs less than 4.7 kb. But within this, these green kind of virus-specific components can be regulatory elements like WPRE. And that WPRE is great for enhancing virus stability and leading to increased expression of your transgene. But as we'll see later, it can cause some complications if you're looking to translate this to the clinic. But what I really want to highlight is our ITRs here. ITR stability is incredibly important and often overlooked. So these ITRs typically come as just standard in your AAV transfer backbone. And they typically are going to be AAV2 ITRs, even if you're packaging in AAV9. Often the ITRs that you're going to use are AAV2. And that doesn't cause any problems. AAV2 ITRs are highly compatible with most serotypes. But the issue arises with these ITRs having a pretty high GC content. So these have around 70% GC, and that can lead to accumulation of mutations. And we recently published a study in nucleic acid research that found the kind of implications of this issue. So we looked at the sort of the sequence identity and how much it matched with records for both 5' and 3' ITRs. So basically, this is what we kind of consider the 5' ITR here. And then up here is the 3' ITR. So we sequenced in particular that 5' ITR because we found that it was very often not compatible with the reported, or not identical to the reported sequence. And just to kind of provide some context for this figure here, when we're looking at our ITRs, we have the sort of plasmid sequence that leads up to this A segment right here, that then leads on to this C and C'. And that's what we're looking at in these sequencing results right here. So this ITR flanking region is what we have in the transfer plasmid. And if we look at the 5' ITR, when we have a GC flanking region that is high in, the ITR flanking region that has a high GC content, we tend to notice a high mutation rate, up to 60% of these 5' ITRs not matching with the reported sequence. And often it's this 11 base pair region right here that gets lost. So what happens here is we have this ITR that's got all of the sort of necessary components, your A segment, your C and C prime arms right here, and it gets shortened into this kind of decreased size ITR, this deleted ITR or recombinant ITR. And when we're looking at just the 5' ITR, this doesn't tend to cause major issues. As long as the 3' ITR is complete, you can still have a recombinant virus that functions exactly as it should. It still does all the great things that AAV does. However, if you have this 5' recombinant ITR or deleted ITR, and you also have deletions that occur in the 3' ITR, that's going to start causing issues with things like full empty ratios. It's going to result in aberrant encapsulation of cellular DNA. You're going to start having pretty big manufacturing and quality control issues. So I noted that this is when you have this very high GC content that you have this high level of mutation rates. Of note, when you have this again, higher GC content ITR, but the flanking region doesn't have that high GC content, you lose that high mutation rate. You increase the stability really significantly. And additionally, when you look at those mutated ITRs or those deleted ITRs, it doesn't seem to really matter about the GC content of that flanking region. You have a pretty low mutation rate either way. But this gives you a kind of cause for concern for the fidelity of your ITRs and therefore the downstream quality control and downstream manufacturability if you are not sure of these sequences and the effect that they may have on your ITRs. So we've actually been working on this really extensively on creating backbone vectors that have these flanking regions that are highly stable that allow for a really high level of stability. If anyone wants to chat about that, of course, we're going to chat. So our ITRs are a point where it's very important to do regular and very thorough QC to allow for our transfer vector to behave exactly as it should. And that's just one of the optimizations that we can make on our transfer vector, in addition to kind of carefully designing these elements and making sure our ITRs have high fidelity, have sequence identity matching with our records, and making sure that this transfer vector has its best chance of success. Additionally, we can make a number of different optimizations on both of our other packaging plasmids, but we're really going to be focusing today on our RepCap plasmid right here. Because not only can you choose a capsid variant that is going to be a serotype that allows you to target the tissue you want, you can also engineer a novel capsid for greatly enhanced specificity. So this leads us to capsid evolution, which allows us to essentially take a capsid, a naturally occurring capsid, and engineer it to have specificity for the particular cell type we're targeting. Now, the problem is that something like, let's say if you're targeting skeletal muscle because you are wanting to introduce the dystrophin gene to, or a version of the dystrophin gene, or the full one maybe, for curing muscular dystrophy, then you have really few choices. It's really just AAV1 or AAV9. But the problem is that those also target a wide array of other tissue types. So what we can do is we can take, let's say, AAV9, And we can modify that capsid through one of these different techniques to try to find an engineered capsid that will only target that skeletal muscle and nothing else. So there are a few different ways that we can create our wide range of capsid structures to screen, including error-prone PCR, which is essentially just introducing random mutations. We can shuffle DNA families, or we can use things like machine learning to try to design an AAV that will perform the way we want. However, the most popular approach is this random peptide display. And this is where we take random peptide sequences, usually between 7 and 9 amino acids, and we place them in a region of that capsid gene that we know is important for regulation or for interaction with our host cell. So this has actually been done before for AAV2. So between position 587 and 588 on AAV2 capsid, previous studies have been done that have done this random peptide display to modify that region that interacts with that HSPG receptor. And we were able to, the scientific community was able to modify that AAV2 capsid in order to better interact with various different host cell types. Now, in addition to better interacting with your specific target and enhancing specificity, you can also screen for capsids that are going to have higher transduction rates, as well as capsids that are able to better evade immune detection. Because that is one issue that we'll talk about shortly with AAV, is that there can be existing immunity that can result in neutralization of your AAV, which is, of course, a matter of pretty significant concern if you're looking at clinical applications. So following, for instance, this random peptide display library construction, these are cloned into AAV capsid plasmids, which are then packaged into various different recombinant viruses and can then be screened either in vitro or in vivo. And this usually takes quite a few rounds to ensure that our hits are actually valid and worth kind of pursuing before we finally identify a novel capsid that we can bring for further validation and further studies. And one thing to note is that these novel capsids will require really, really extensive study. So these novel capsids are a great way to target very specific cell types. But in addition to this, we can also enhance not only specificity, but also optimizing the payload of our transfer vector by engineering different specific promoters. So the first step is pretty similar. You're going to create a pool of various different promoters. This can either be through very, very specific design, designing each and every promoter variant, or we can use directed evolution, which is kind of or we can use target enrichment for genomic DNA, which is a more unbiased approach. Essentially, you take genomic DNA fragment, you can use probes to kind of isolate a little bit more variants, and then you screen essentially all of those variants for almost like naturally occurring promoter activity. Or you can kind of take an in-between approach, and you can use directed evolution, that is things like family shuffling, and more specific modifications to those existing promoters. From here, you're going to construct a library. For instance, you have your promoter variant, which is upstream of some reporter. You have your barcodes so that you can actually identify which promoters are behaving the way that you want, followed by a poly-A2. And then after screening, you're going to go through this same, again, kind of cyclical screening, hit identification, and then kind of further screening to hopefully narrow down that pool into a small number of candidates for validation. And we have actually performed this process specifically looking at cone cells, because the existing cone-specific promoters, especially Pro-A1, which is the most popular, are very large. They're mostly around 2 kb. So if we have that 2 kb promoter, we have very, very little room for any therapeutic genes that we can deliver to those cone cells in the eye for treatment of various different genetic disorders. And these therapeutic genes are by and large pretty big. Some of them are smaller, around 250 base pairs, but others reach well over 3 kb, which puts them out of the range of possibility for our standard sort of AAV backbone. So screening for a decreased size promoter will leave a lot more space for therapeutic genes and allow just a lot more flexibility, a lot more optimization. So here we took the Pro-A1 promoter as well as a couple of others, and we went through this promoter engineering process, making modifications to that Pro-A1 to try to identify a much smaller promoter. So we found a wide array of variants, we packaged them into our recombinant virus, and we performed this subretinal injection and analysis. And through a few rounds of screening and a lot of work by our R&D team, we were able to identify a novel cone-specific promoter that's only 458 base pairs. So it's around a quarter of the size of that stereotypical Pro-A1 promoter, leaving so much more room for therapeutic potential, basically, in those AAV transfer vectors. So after you kind of select that promoter, especially for things like specificity, the job isn't necessarily done. I do want to emphasize that it's important to not really stop there, because beyond just selection, there is a huge amount of potential for promoter sequence optimization. Here's one particular case study where we took a mutant for an ORF that results in high lethality compared to the wild type, and we took that same replacement ORF and put it downstream of various different sequence optimized versions of the same promoter, because not only do you need to have sequence specificity, but you need to monitor that transgene expression. And as you can see, there is wide variation in the amount of transgene that is expressed, especially over time, for these different versions of this promoter. So that sequence optimization following identification is really important, not only for kind of these novel promoters, but it also can be really beneficial for natural promoters as well. And before we kind of wrap things up, because I am close to the time that I wanted to leave for plenty of questions, I do want to note a few key considerations for clinical applications. So when we're looking at whoops, that went away. Let's see. There we go. Okay, hopefully everyone can still see this. So when we're looking at clinical applications, one of the biggest things to consider is potential safety concerns. And first and foremost, I mentioned that antibiotic resistance marker that we need in all of our plasmids. Ampicillin is the most common, but this has to be changed first and foremost if you are planning on getting to the clinic eventually. The sooner the better for changing this is great. So you either want to change that to kanamycin, or you can use a vector, a miniaturized vector that doesn't have any sort of ampicillin, that doesn't have any antibiotic selection marker. So there are a wide variety of different options for this available, these miniaturized vectors. We have our own named MiniVec that doesn't rely on any sort of selection markers, but these are great options for clinical development for not only your transfer vector, but also your packaging plasmids as well. Additionally, you need to check those regulatory elements. I mentioned this before with our WPRE. You want to avoid the wild type WPRE because this is a viral element. And so it's going to cause issues with any sort of FDA regulation approval. So there are a couple of other options that you can use for modifications to that WPRE, but really generally, using any untested elements is very, very tricky when it comes to just general, any extraneous sequence that doesn't really need to be there, especially anything that's untested, should be removed. But also, you have to be aware of your entire vector, including those novel capsids, including novel promoters. All of those things need to be really, really thoroughly tested well before clinical phases, ideally even during preclinical phases. The earlier you can start these considerations, the better. And one thing that we're very, very big on is this good vector practice, basically making sure that your vectors are designed for the end goal from the very beginning. And so that's taking into account things like safety concerns, but also manufacturing. We want to be sure that we have that ITR stability, and we want to make sure that all of our other sequences are highly stable and aren't going to be prone to things like recombination or cloning issues. And of course, we want to make sure that whatever we're making is efficacious. So test everything. And as I mentioned before, with AAV, there is the risk of those neutralizing antibodies. That's one reason that capsid evolution tends to be pretty popular. It's because we want to try to identify a capsid that won't even be recognized by the immune system. But there are a variety of tests that you can do to check how big of an issue that would be for a naturally occurring serotype. And so you can consider pairing one of those engineered capsids with an engineered promoter for really effective performance. But that's going to require a very, very thorough amount of testing. And so that's something to be very, very highly aware of. So in summary, AAV is a great system if you're looking to deliver genetic material in vivo for either research or clinical purposes, if you're looking for long-term non-integrated expression, and if you're targeting your transgene or your RNA to a particular cell type. So designing those transfer vectors is going to require really careful consideration of your promoter size, your promoter type, and of course, your ORF needs to be well matched to your promoter, and it needs to be within the appropriate size. You also need to look at things like your regulatory elements and your ITR stability. So you can make a huge amount of optimizations, way more than we have time for today, including that capsid and promoter engineering. But there are a wide array of other options as well that we can talk more about. And I encourage you to consider clinical applications very, very early. Too often, we at Vector Builders see people coming in looking for clinical manufacturing of vectors that have not been fully optimized. And any changes that you make for, let's say, safety or manufacturing are going to potentially affect your efficacy. So these are things to consider early to basically save yourself the headache in terms of time and money later on. So thank you so much for your time. I greatly appreciate it, and I'd love to take any questions.
Thank you so much, Connie, for that very detailed presentation. That was fantastic. We do have some questions. The first one is it possible to exceed the 4.7 kb by a certain percentage and still get a decent packaging efficiency? What about the minimum size below which AAV production won't be efficient? Production or functionality won't be efficient?
That is a great question. So the short answer is you can exceed that 4.7 kb. That is possible. Just like for any vector system, you can theoretically clone and package something bigger than the maximum capacity. There isn't really a very highly defined point at which the titer of your virus is just going to drop off a cliff. That's going to be dependent on a lot of different things, including just your actual sequence that you're packaging. Some sequences are more difficult just in general, but then the bigger you make them, the more you're going to affect your titer. So I'm sorry that the answer is it depends, but really the answer is it depends.
Thank you. So for muscular dystrophy, can that gene be replaced adequately?
That is an excellent question. Currently, I mentioned that a version of the dystrophin gene, I said something about that when I talked about muscular dystrophy. That's the currently approved AAV because the dystrophin gene is really big. And so you cannot just replace that on its own. There is a mini dystrophin that can be introduced with AAV, but the success is a bit more variable. Ideally, you want to replace as close to the natural gene as possible. And actually, we have been developing a system by which you can do dual AAV introduction, where two AAVs each carry part of a larger protein that can then go through recombination to create the larger protein on its own. And we've been seeing some really promising results.
Fantastic. Can you comment on making AAV split designs with recombinogenic signals to allow for the expression of larger genes that might otherwise exceed cargo capacity?
So yeah, that's actually exactly that. So really just it requires a huge amount of optimization and has actually been going on in our R&D for a very, very long time now, trying to figure out how to make this work very, very consistently. But yeah, essentially you take two segments of the protein, potentially more, but let's say two segments of that protein that can then recombine in the host cell into a fully functional large protein.
Very cool. Thank you. We have a question about capsid engineering. Could you give a practical example how to enhance the targeting with capsid and a promoter engineered, and if that is also used in mice?
So yes, it is used in mice. There we go. There we go. I'd like to have a visual aid. So yes, so for in terms of a practical example, AAV, that AAV2 example where random peptide display of that HSPG binding domain what is kind of a classical example of just a pretty straightforward way. Like it's quasi low hanging fruit, I guess, of you know exactly where it binds, you know where they interact, modify it until you can get the targeting that you want. I'm so sorry. Can you read the second part of the question again?
Yes. Um, and if that is also used in mice? Was that?
Sorry, maybe the part before.
Yeah, practical example of how to enhance the targeting with capsid and a promoter engineered, and if that is also used in mice.
Yes, thank you. Yeah. So for the capsid, exactly. Yes, we can definitely use this in mice to enhance targeting. There we also have various platforms for making sure that this works across multiple species to just increase the kind of translatability. Because of course it's an investment and you want to make sure that this works in a variety of systems if you want to end up taking this capsid to the clinic. So yeah, we can have our capsid that can be engineered. Again, that random peptide display is the most popular, but we have a variety of other options as well. But essentially, after introducing to your target cells, let's say you introduce it to a mouse in vivo and you're trying to target, just for the sake of the example, skeletal muscle, you can then isolate the skeletal muscle initially if you're just caring about optimized targeting there, or you can do a wider array of sampling if you're interested in potential off-target targeting. And you can isolate, you can check for those barcodes, you can do either DNA or RNA sequencing. We have a lot more information about that for the screening process, and then go through multiple rounds to eventually identify your main candidate. And really, it's a very similar story for promoter engineering. Both of these are library screens where you're taking something that works okay, or you can potentially take a bit more of a kind of shotgun approach. And so you're taking either something that works, or let's say the entire genome, and you're screening for something that's going to target specifically what you're looking for.
Thank you, Connie. We have a question about the cone-specific promoter data that you presented. They're wondering if we are planning to publish it or if it's already been published.
So this has not been published yet. So it is hopefully going to make its way into a paper soon. You can find this data actually on a couple of different places on our website for now. That's really the only place that it's published currently. But yeah, to be determined.
Thank you. We have an interesting question. I'm sure a lot of you are wondering this. Is there an AI software to help testing my designed vector for working well? This is because I'm curious whether a vector will work like pretest as I intend it to.
That is an excellent question. So interestingly, there was a conversation on, I think it was LinkedIn, about this, about basically the utility of AI. Can AI just go ahead and design gene therapy for anything right now, please? That would be lovely, wouldn't it? However, really, AI is as good as its training, right? And basically, the amount of training that can go into AI depends on our understanding as a scientific community. And there are still so many question marks about how any particular vector is going to work. We can make our best guess, of course, and we can avoid some common pitfalls like mismatching of an ORF and a promoter. But really, to determine if your vector is going to work exactly as you intend, it requires that testing in the lab. And of course, there are options for pilot tests, for small scale tests. There are checks that you can make along the way. And actually, the design team at Vector Builder have seen just about everything. And so they are really good at picking out errors. This is an instance where AI doesn't have the same experience that they have in our experience, because we have tried out a variety of different ways to use AI for vector design, and we continue to find a lot of errors. So really relying on the experts is still the best option.
Thank you. Could you please elaborate more on the potential immunity-related outcomes of AAV?
Absolutely. So in terms of, I'll talk about the unintended immunity issues, because I think that's what we're talking about here. So in terms of an immune reaction, AAV has the lowest immunogenicity, far lower than adenovirus, which tends to cause a pretty significant immune response. So really, in terms of off-target effects due to immune reaction, super low risk. The more kind of pertinent issue, especially when we're looking at clinical applications, is going to be more related to potential silencing of your AAV. Essentially, if someone has been exposed to that particular serotype before, they are going to have existing antibodies for it. And so that AAV is probably going to be silenced.
Okay. We have another question. How can we overcome problems with instability of ITR in AAV production?
So the multi-layered, of course, as always. Really the biggest thing that you can do for ensuring ITR stability is watching that GC content. That's the thing in terms of the data that we are seeing to the largest degree. Is that if you have high GC content, not only in your ITRs, but also in the flanking sequences, you're going to have low stability. I mentioned that we have been actually working on an AAV backbone that has very, very, very high stability. We've been doing our own library screens of all of these ITRs and their flanking sequences. And we have found a particular sequence that is so stable that it has not resulted in any mutations over hundreds of rounds of replication of this virus. And I'm pretty sure it's far more than that, but I'm not confident enough to say that at this point.
All right, thank you, Connie. We have another question about what is the best way to test the off-targeting of AAV in our model in case we're using it in vivo.
So in terms of, I think you blipped out for a second there. I'm so sorry. Can you repeat?
Yeah. What is the best way to test the off-targeting of an AAV in our model in case we are using it in vivo?
That is a great question. So I would say starting with knowing the most likely sources of off-target effects is a great starting point. So if you're using an AAV serotype that targets a wide variety of tissues, take a look at those tissues. So doing things like biodistribution profiling. We have a lot of experience with that, and we've seen a lot of instances where depending on especially the sequence of the AAV, you can have pretty surprising distribution among the known targets. Just because an AAV targets one particular tissue or another doesn't mean it's going to target it equally, right? And so checking all of those different target tissues through things like biodistribution profiling is going to be a really, really good idea. Of course, ideally you would test every tissue type, but if you know which tissues your serotype is targeting, you can start there, and that's a great place to look.
Great. We have another question. Do you think AAV is the most promising gene delivery vector currently for all therapeutic applications?
Oh, oh, I, I love that question. It's a very hard question. Um, I would say it very heavily depends. I'm so sorry. I hate saying it depends, but it depends on your particular application. I would say for if we're talking about like delivery in vivo, let's say neurodegenerative diseases, AAV is my bet for sure. Ocular diseases, AAV for sure is my feeling. If we're talking about things like vaccine delivery, there are probably other options that might end up beating out AAV. So I mentioned that like non-viral vectors are incredibly useful in certain contexts. Vaccine delivery is a great example because you don't need super long-term expression. You can just express that antigen short-term, allow your host, your person, to develop an immune response to that and develop antibodies against that antigen, and then you're done. So you don't need it for that long. But if you want something with sustained expression, if you want something without the risk of host genome disruption, AAV is just a really, really good option. If we're talking about cancer treatment, if we're talking about oncolytic viruses, if we're talking about vaccine development, that's a much trickier subject, but I think there are some areas where AAV is a clear winner.
Fantastic. That was a really well-explained answer for a complicated question. And in the interest of time, I think we will end our question and answer session right here. But if you have more questions, feel free to contact us. We'll be happy to answer any questions that you have. And I want to thank all of you for attending this seminar. Thank you very much. And we hope to see you in our next seminar. We will be publicizing that. We will let you know when that is. Thank you so much, Connie.
Thank you all very much. And Samita's computer might have frozen, so I'll go ahead and sign off for both of us.
Oh,
Thank
okay.
you, everyone.